CAR T therapy works. When it works, response rates hit 80 to 90%. The problem is the process. Today, treating one patient takes weeks of manufacturing, around $500,000, and a stay at a major clinical center. It works, it just doesn't scale. My guest today is Dr. Salim Dhanji, CEO and co-founder of ME Therapeutics in Vancouver, British Columbia. He's an immunologist with 25 years in the field, and he's working on something that could change the entire model, an in vivo CAR. Rather than extracting a patient's cells, engineering them in a lab, and reinfusing them, you reprogram the immune system in the body with an injection. We get into the science, the path to first in human, and what it takes to raise capital for Biotech in Canada in 2026. I'm Christopher Wilson. Welcome to Stories in Life Sciences. Let's get into it.
This is Stories in Life Sciences brought to you by MedAxis AI, where we explore how ideas move from discovery to real-world impact.
I know a little bit about this area. I'm excited to dig a little further in. It seems like some exciting things that you're working on in the immunocancer sector up there in Vancouver. Just to start off, that's where you're calling in from today?
That's right, in Vancouver, Canada.
In Vancouver? Okay. Yeah, and just to start off with, I always like to know, just from a guest, like just your arc, like what led you to starting ME Therapeutics and some of your background?
I'm an immunologist by training, so I've studied the immune response to cancer for the last 25 years. Did my PhD out at University of British Columbia, did a fellowship out at the Princess Margaret Cancer Centre in Toronto, really just understanding how the immune system can function to fight cancer, but also the opposite side of that, where how the immune system plays a role in autoimmune disease, how it's almost like two sides of the same coin. My background has been really focused on developing drugs that can reprogram immune cells to really fight cancer and treat patients who weren't previously treatable.
Yeah, and so that's what I was reading about too. And now, so the therapy that you're working on, the CAR therapy, like how does that work now? And then how is your therapy different than that, how that functions now?
One of the interesting areas that we've been working on in the last couple of years has been what you mentioned, this CAR therapy, which is actually, CAR stands for chimeric antigen receptors. It's actually a really
CAR is easier. I know what I abbreviate, like the acronym is there.
Absolutely. So CAR therapy, what it is actually people can now program immune cells from a patient to actually express this CAR receptor, which directly recognizes their own cancer. So you can then grow out these cells, engineer them to express the CAR, and then reinfuse them into patients. And you get these immune cells expanding and actually going in and finding the cancer cells and killing them. It works really well with certain blood cancers, and that's really where most of the current CAR therapies have been approved. The challenge is that the approach requires you to have a patient come in who has late stage cancer. You have to isolate their immune cells. You have to grow them in a dish. You have to engineer them, and then you have to reinfuse them into the patient. So the whole process can take anywhere from 4 to 8 weeks and requires the patient receiving chemotherapy and then reinfusion of the immune cells back into the patient. So the whole process takes a long time. A lot of these patients are really sick, and so they can't make it for that 4 to 8 weeks. And on top of that, they have to move to a clinical center where these types of treatments are available. You can't really treat these patients outside of these major centers, so they have to figure out a way to come and live there for 3 or 4 months and get this treatment. So it's a really challenging approach, but it's been very successful. So when patients have been treated with this type of therapy, you're seeing response rates of 80 to 90%, where the patients are more or less cured of their cancer for that time. Unfortunately, some of those patients will relapse. One of the challenges that we're trying to solve, but given the complexity of this manufacturing process and the time it takes, the way the field is moving now and what we're doing now is actually trying to do all of this reprogramming in the patient with a simple injection of a drug, rather than having to isolate the patient's cells, do everything outside the body, and then reinfuse it. And so we're starting to see a success in that area, and we're really hopeful that by moving to this, what we call an in vivo CAR setting, we can get more patients treated and be able to treat a bigger patient population.
Yeah, and I assume what I've seen, it's also quite costly to do it, the current form that they're doing it in.
That's right. And right now, I think it's close to $500,000 per patient.
Yeah. Wow.
for this type of treatment. And obviously, trying to get those costs down is quite important to be able to make it accessible to more people. The approach of moving to in vivo, I think, will help to solve some of those challenges.
And how are you tackling that? Or when did you see, okay, this is what's being done now, this is what we could do, and then how are you going about solving that?
Part of it stemmed from kind of the COVID vaccine time and COVID time, when you started to see the ability of delivering nucleic acid based drugs to patients. Vaccines, for example, during COVID, you could now safely administer nucleic acids and reprogram cells in a patient using lipid nanoparticle technology, which delivers nucleic acids to immune cells. More importantly, because we were working with nucleic acids as a drug now, you could basically go from an idea to having a testable drug in weeks rather than, months to years, which was the normal process. And so as a scientist, we saw this opportunity as, how do we take this technology, which is now mainstream, and apply it to some of the challenges that we had in the cancer setting, for example. And that's really the approach that we've taken. What we're doing is we're actually working with kind of world leaders in the nanoparticle space so that we can really specifically deliver our cargo, which is these CARs, to immune cells in a patient. And our lab is really focused on optimizing this process and trying to get into a clinical trial as soon as possible.
Wow, I was looking at how the delivery mechanism is one thing, but once it goes into the tumor, just if you could speak a little bit more to that, like what's actually happening, like inside the tumor, I feel like is just such a complex place to that. I feel like I like didn't really understand the full level of complexity with it. So once the injection happens and it occurs, it's waking up our own ability to fight back, which has been suppressed essentially. Is that kind of?
Yeah, so it's kind of a dual approach. So the CAR therapy is actually taking your immune cells, and instead of them recognizing, the virus or the pathogen that they would normally recognize, you're actually putting in a receptor, the CAR, that directly recognizes some protein on your tumor. And by doing so, those immune cells are now armed that the minute they recognize that protein on the tumor, they actually kill the tumor cell. The CAR is protein-based receptor. And so what we're doing is we're delivering the nucleic acid, which encodes for that receptor to immune cells in the patient's body. And so they're not necessarily in the tumor. They could be circulating outside of the tumor, but once they express the receptor, they naturally go in and find the tumor cells, and then they get activated and they kill the tumor. So that's, one of the approaches that we're taking. And the other thing, which we haven't really touched on, which is the tumor microenvironment, which are also quite focused on, or CAR therapies in general are effective for blood cancers in general, because they're circulating, they're easier to access by immune cells. Okay. But when you have solid tumors, for example, they're much more challenging because the tumor microenvironment is not just tumor cells, it's actually other supporting cells that are part of your body, immune cells, which are actually guarding the tumor and suppressing your natural immune response. And so for solid cancers, which we're also developing therapies for, this suppression in the tumor microenvironment and these immune cells that are present that are suppressing are an important target, because if we can overcome suppression by those cells, or if we can eliminate them from the tumor environment, we can more or less open up the tumor for attack by the rest of your immune system. And so we have a program where we're actually using the same nanoparticle type technology to actually deliver nucleic acids that target important, like scientifically validated pathways that everyone knows are key to creating a strong anti-cancer response and reducing immune suppression. In the past, those targets have been difficult to go after because, you get systemic toxicity in some cases, or changes in the tumor that don't allow your drugs to work. But, with this nucleic acid technology and the delivery technology we're working on, we're actually getting, we're working on specific delivery of specific targeting of these pathways in the tumor environment and not in other places. So you kind of overcome some of the systemic effects. And so a lot of these pathways that we know are important as scientists, we're now able to go after with this new technology.
Wow. And then at what point were you in the lab, or whatever that moment was, that you saw, okay, this is what's being done now, this is what you're finding, to then I'm going to start a company?
Yeah. So I guess I was in the academic space, I did a fellowship out in Toronto. Right after that, I actually moved to a startup company, and I was their second employee. It was a biotech company focused on cancer immunology. And really, this was, around 2009 when the first drugs in the field of what they call immuno-oncology or immune-based drugs to treat cancer were actually being approved. So in the early 2011, 2012 timeframe, you had these immune checkpoint inhibitors, they call them, that were
Why
being
was it
approved.
then that they were getting approved? Was there a certain thing that happened or technology that was available or what?
No, so what happened was, that's really when the first clinical trials were
Okay.
completed. What we realized was that the immune system did play a very important role in fighting cancer, and cancer had developed a way of suppressing the immune system. And if you could find drugs that could block the way the immune system was being suppressed, you can actually unlock the immune response. And so these checkpoint inhibitors, they call them, they targeted one or two different pathways in which T cells, which are your cancer killing cells, are suppressed. If you could block those with these checkpoint inhibitors, you could see that a patient's own immune cells could go in and fight their own cancer. It was really exciting because you had patients who had very late stage, stage 4 kind of metastatic disease that had spread throughout their body. Some of them were, I just heard a story today about one of the first patients treated with this therapy. They had, metastatic disease that had spread all over. They were getting ready to go to hospice. They got onto one of these trials and were treated with these immuno-oncology drugs. That patient is still alive today. They
Wow.
had such a remarkable response that, 15 years later almost, that they're still here when they were supposed to be gone, 2 months after they were supposed to get treated. Those are the kind of stories that are amazing. Those checkpoint inhibitors, for example, at that time, we realized that they do work, but they don't work in all patients. And so there was a lot of immune suppression that was happening that prevented some of these drugs that worked in a lot of patients from working. And so the response rate to these checkpoint inhibitors is depending on which cancer it is, it could be 5 to 50% of patients will
Well,
respond. And the exciting thing is it works across cancers. So you could treat a patient with melanoma or skin cancer, or you could treat a patient who had lung cancer with this same drug. And because you were targeting the immune system, you could actually treat both types of cancer. The field is really exciting, and the applicability of these types of treatments was so broad. But again, the response rate was the challenge, right? So
how is that so widespread? Like 5 to 50%?
in a lot of cases, it depends because these treatments depend on your immune system naturally recognizing the cancer. So if you had an underlying recognition of your own cancer, but it was being suppressed, you could overcome that suppression and your immune system would go in and fight the cancer. But in some cases, there are other levels of suppression. And in other cases, there's just not enough changes in your cancer for your immune system to recognize them. So there was kind of multiple challenges, but one of the bigger ones was really these other levels of suppression. Given my background as an immunologist, we realized that there were certain cells in the tumor microenvironment that were playing a very big role in preventing patients from responding to these immune-based drugs. And if we could target those cells and either eliminate them or create a better anti-cancer tumor microenvironment, we could allow those drugs to be effective in those patients as well. And so that was really the reason that ME Therapeutics was founded, was to go after the tumor microenvironment and be able to reshape it so that you could create a strong anti-cancer response and allow more people to benefit from these types of therapies.
Sounds like a simple problem to tackle.
Yeah, it's turned out to be a lot more challenging. I know with technology now, it's become easier. One of the things that, like I said, what we're working on is this very specific delivery of nucleic acids to the tumor microenvironment so we can specifically target these immune cells and create a stronger anti-cancer immune response. And then on top of that, if you can reprogram your immune cells to actually recognize your cancer, you can kind of hit it from both sides. Hopefully, we can treat patients that were not treatable. Unfortunately, I have a good friend who was diagnosed, he's 40 years old, diagnosed with stage 4 kind of glioblastoma. And you look at it and the only treatment right now is chemotherapy, right? And radiation. And then it's a kind of a wait and see approach. And, it's been the same treatment for 20 years or more, right? In a lot of cancers. It's about time that we're able to tackle some of these challenges and some of these cancers with new drugs because we have the technology to do it now.
Yeah, no, that's exciting. That's a
Yeah.
reason to keep keep going, I'm sure, during the challenging times. And
Absolutely. Yeah.
what's that been like? I mean, making that transition from science to business.
It's been fun for the most part, aside from the
That's good.
constant challenge of financing the company.
Yeah.
But, I've always been entrepreneurial. I see opportunity to apply, my scientific knowledge to running a successful company. I think we see how we can approach things a little bit differently, but also, being able to communicate that with investors and really say, this is why we're different. This is, what's exciting about what we're doing. It's been a challenge, but it's been a fun learning experience, to be honest with you.
And you mentioned that. I mean, most of that's you've really enjoyed, and you had that entrepreneurial spirit. I find not that can be a challenge for different people coming from the scientific or academic environment. But it seems like you already had that startup environment experience prior to starting Me as well.
Yeah.
But what's really surprised you about it, or anything that's like been different that you thought it would be to go out on your and create your own?
The thing I underestimated the most was really, especially being in Canada, just the funding environment being a lot more challenging than I had expected. We really feel the need now to get a broader investor base and move out to the US at least to be able to access some investors and capital that's not available here. The financing side has been the biggest challenge, to be honest with you. I think we have great science and we have, great people, and, we're very lean and able to do things cost effectively. We've been able to stick around with the limited financing that we've had. But, once you start getting into clinical trials, without the funding, the large amounts of funding you need, it's impossible. So really, from a company perspective, we realize that, we need to be able to access that broader investor base. And, that's part of our kind of corporate strategy moving forward.
Yeah, and I noticed you were at JPM this last year, correct?
Yeah.
That was my first experience there. So,
Right.
but I heard from a lot of people that they felt, I don't know, more optimistic after leaving that meeting this year. Like there's more capital coming in, to the market. How did you feel, or what was your experience there with your conversations with me specifically too, with investors? Or what was the perception?
Yeah, absolutely. It was definitely a big change. The last 4 or 5 years have been quite challenging in the investor side in life sciences and biotechnology. What you're seeing is that, over the last year, you're starting to see glimmers of hope where the biotech index has started to be on the uptick again. You're starting to see money flow. You're starting to see deals happen from big pharma companies purchasing smaller pharmaceutical companies. JP Morgan, we realized that, this year people were starting to feel hopeful.
Yeah.
I think we're continuing to feel hopeful, which is nice. You are still seeing deal flow, which is exciting. And we're in an exciting area of biotechnology with this in vivo immune reprogramming and in vivo CAR space, where you have seen a lot of deals happen in the last year for sure. So we're excited about that. We want to keep pushing forward.
Where are those at for you all? I know you have a couple of things that I look at that you're working on or what's in the pipeline, but do you want to speak a little bit more to where those pieces are and where you're at from what's closest to human trial and just overall?
Yeah, our lead program is this dual CD19, CD22 targeted CAR. It's an in vivo CAR that we're developing to treat patients with cancer, certain types of blood cancers. In the last year, what we did was we licensed a technology from the Canadian government, from the National Research Council of Canada. It was a CD22 based CAR. So CD22 is a target on certain blood cancers. That CD22 CAR currently is being tested, not by us, but it's being tested in a clinical trial. We own the rights to that CD22 asset. The nice thing is it's being de-risked in the clinic as we speak. But what we want to do is we're taking that CD22 and we're moving it into the in vivo setting. What's being tested right now is kind of the old school, the original version where you have to actually reprogram a patient's immune cells outside their body and then reinfuse it. We want to take that as asset and move it straight to an in vivo setting. And so we're developing this in vivo CD19, CD22 CAR to treat cancer patients with a simple injection or multiple injections versus this ex vivo manufacturing process. And right now what we're doing is we're just optimizing our lead. And the plan is to actually move it into animal testing over the next few weeks to months. And then move it into a clinical setting, hopefully in the next year and a half or so. That's kind of what we're hoping. Part of the timeline depends on us raising the financing to start the phase 1 study. So unfortunately, it's very costly to manufacture a drug to treat humans, to do all the testing that's needed before you can even inject your first patient. So, we're looking to raise that money right now and expedite the process as much as we can.
Yeah, no, that's exciting. That would be extremely disruptive, it feels like, too. Just
Yeah.
a lot of pharma companies in that world. I mean, if you're changing that life cycle from months to getting that therapy to doing it in vivo, I feel like there's probably a lot of money that's into the manufacturing and the creating of how the process is now to what you would be looking to move to.
For sure. And our approach has always been to work with experts in the area of this formulation and drug development side. The nanoparticle is one of the keys to getting this in vivo CAR to work effectively. Yeah. And we're surrounded by the world experts in this technology. And so we really rely on them to give us their best formulations for this approach. They'll help us with the manufacturing side and helping us get the drug manufactured to a point where we can actually treat our first patients. And so it's really a partnership with, local experts and local companies that are experts in that field.
Yeah, I feel like nanoparticles have evolved so much. I was in school like 10 years ago. I feel like I was looking at them and then I turned away and came back, and I'm just blown away by what they can do for delivery systems. And it's unbelievable now.
Yeah, absolutely. And some of the most exciting stuff is you can now target these nanoparticles to certain immune cells, right? That the first original nanoparticles, when you injected them, the majority of them are actually delivered to the liver, and that's kind of where they get soaked up, so to speak, right, and cleared from your system. And then you had nanoparticles that would stay in circulation longer and avoided being taken up by the liver for a while, and that allowed those nanoparticles to kind of get out of the blood and into tissues where there were leaky blood vessels, like in cancer. Then it allowed those nanoparticles to accumulate in those cancers. Now you can actually take those particles and you can modify them in certain ways to actually make them specifically or at least enhance uptake by certain immune cells or certain other cell types, rather than liver cells or cells that you don't want taking up the particles. So there's a lot of engineering that's happening and that's happened. And there's a lot of experts in that area. And I don't pretend to be one of those people, but we rely on them to help us with their delivery technology. And the things that are coming out over the last couple of years have been really exciting and really unlock what we're trying to do.
Yeah, it's a good perfect storm of these different pieces coming together to allow us to really advance in this space.
Yeah, absolutely. And it's not like, we just started working in the area. We've also, when we first started the company, we've realized that lipid nanoparticles were an effective way to actually target the tumor microenvironment. Aside from the liver, which I mentioned, naturally takes up these particles, there's certain immune cells called myeloid cells, macrophages, that make up a big part of the tumor microenvironment. Mm-hmm. And they're naturally phagocytic, or they naturally take up foreign particles from the blood. And so one of those things is they are a natural sink for things like lipid nanoparticles. And so we said, okay, if we can formulate drugs into lipid nanoparticles, and we know those nanoparticles are naturally taken up by myeloid cells that make up a big part of the tumor microenvironment, we can use that system to really deliver drugs specifically to the tumor and have an impact there. And so that's kind of where the original thought process was. And at that time, we were really looking at, what they call small molecule drugs or chemical-based drugs. But over the last 3 or 4 years, like I said, given the advances in nucleic acid technology, you can now deliver these nucleic acids into the tumor and let them encode for whatever drug you want it to encode. And you can activate pathways or suppress pathways in the tumor specifically that from a scientific basis are going to be key for generating a strong anti-cancer response. That's really where we focused a lot of our attention over the last couple of years, for sure.
Wow. Yeah, I wish I could watch what's going on, like what's happening there.
It's exciting. And you can kind of take a lot of these studies, you can take these particles and inject them into mice, for example, that have cancers, and you can image them and you can show that, look, they're accumulating here. Your immune cells are changing and you're getting an anti-cancer immune response. As a scientist, that's always the most fulfilling part of your job, right? When things are happening. And it's exciting to be the first person in a lot of cases to actually observe something like that, right? An anti-cancer response with your drug and, your hypothesis working, hopefully not working.
Yeah.
Yeah.
No, I work with, I'm going there later today. They do spatial biology, a CRO here in Portland, and the imaging, they're testing a lot of new drugs like this. And it's really,
Yeah.
yeah, it's amazing to see some people working on things like you, and the ability for the first time for them to actually see, wow, like this is what's
Yeah. Yeah.
happening.
It's so, I mean, I've been out of the lab for the last, 10 or so years, I guess now. The last time I was actually hands-on on an experiment. The technology that's available now for even for imaging, like we can, what we used to have to do is take, start a study and then pick a fixed time point and say, okay, this is where I'm going to look to see if it's working or not. Now you can image, what's happening over time. So you don't have to say, okay, I'm going to look 1 hour later. You can actually look at 1 hour, 2 hours, 3 hours, 4 hours, because in a lot of cases, things don't happen on the timeline that you expect. And now with the imaging technology and everything that's brought costs down for research, that you have this ability to look over the longer term and see things that maybe you wouldn't have seen before. Even for non-scientists, it makes a difference to be able to see what's happening. Right? So if you can say, okay, this is the cancer, right? This is what's glowing here. And now it's not glowing anymore, right? After we treat it. So it's very black and white. So you can, it's easier for people to understand. And from a company perspective, like for investors, it's, that's the approach that we try to take, is make it simple so that people can see what's actually happening, right? Rather than needing the technical knowledge to understand it.
Yeah, no, definitely. I was like, it's fun. I'm super visual too, and walking around in their lab and what they're testing on to see the biomarkers and what they can actually show and how it's attacking it. It's really helpful to be able to see that. And they've said that some companies, it's helpful for them from an investment perspective to just show some of the visuals of like, this is what's going on. This is our latest round of testing, and can see it lit up on the screen of what it's targeting and what it's doing.
Technology has come a long way, right? It's really unlocking the future, and you're going to see a lot more personalized therapies and kind of single patient therapies that will come down the pipeline. Cancer vaccines, for example, have been shown, and without getting, it's not something that we're really working on, but it's exciting because cancer vaccines are not your typical vaccine where they prevent cancer. You actually have patients who have cancer. We can sequence their cancer. We can make a personalized vaccine to actually amplify their immune response to their own cancer that's already in the body. And you're starting to see patients, responding to these types of treatments. These are really personalized treatments. And it's exciting because, you don't have to rely on, one drug that's going to work for everyone. You can modify it for specific people and hopefully be able to generate anti-cancer responses and cures, hopefully, for patients who didn't have any cures before. So
yeah, the diagnostic, the personalized medicine, that is really exciting. That aspect of it to me too, the testing that's able to be done, that wasn't able to be done before, and where that's going. You mentioned earlier you haven't done a test or been in the lab in a while, and just I've talked to a lot of scientists coming from that. What's that been like, that transition? Do you miss it, or what's that transition like of going out of there completely?
I do definitely miss it. I'm not going to lie. I enjoy the science part of my job a lot more than, the business side.
Yeah.
The financial side is great. I know I have to do it to finance what we're doing, but really the exciting stuff is the science. And technically I've always been pretty strong in the lab. So I've always enjoyed doing experiments and coming up with new ways to test your hypothesis and actually seeing the results as they're happening, right? So, that's always been an exciting part of what I do. I still get to do the big picture science, right?
Yeah.
And planning and saying, okay, this is what we should do. This is how we should approach it. And then relying on our team to carry it out. And as much as I miss it, I don't know if I could go back and do that stuff anymore. Given my experience now, the big picture understanding is where I provide the most value, right?
Yeah.
And the technical aspect can be carried out by the younger people in the lab, I guess.
Yeah. And you mentioned the team part. Like, what's that aspect of building a team, and any advice that you have on that, or how did you look at creating that atmosphere?
Yeah, I've always approached things, somewhat differently. Like I'm not, I guess when you look at biotechnology companies or science-driven companies, you end up with a lot of people with PhDs, and that's kind of the bar that you need to be a scientist level position. We've approached things a little differently in the sense that I don't necessarily need you to have a PhD as long as you can prove to me that you know what you're doing and you're capable and you're willing to learn. Those are the real people that you want working for you. Obviously, you need the PhDs as well because they can understand, the big picture of what you're doing and guide some of what you're doing. I don't think it's necessarily a prerequisite when we built the company. It's really been hiring people that are driven and motivated to do the work, realize that we're trying to do something bigger and try to treat cancer and patients that weren't previously treatable. And it's been important to me to find hardworking people that work well together and are motivated, but obviously technologically capable of carrying out the studies. So we've been lucky to have a lot of those people around, and it's worked out great for us. So
yeah, that sounds exciting. And then for you, what's next, or what are you most excited about, I guess, where you're at right now and what you're working on and the evolution of the company and where you're going?
Yeah, I think the most exciting thing is really trying to get our first in human study going. That is our main focus, and really part of that is articulating the story and telling people what we're doing and why it's important so that we can raise the funds that we need to get our first in human study going. Our approach has been unique in that what we think is a significantly de-risked asset, this in vivo CD19-22 CAR, because the 19 and 22 components have been in humans before in another format, right? So I think, from a safety perspective, I think we've de-risked the program. Getting into humans with our in vivo CAR provides a lower safety risk, and we know that those CARs work in another format. So we would think that they should be efficacious. And so we're really excited to prove that out, really, and get this into patients as quick as possible.
Next step in people.
Yeah, exactly.
What is your current, you're actively raising for a next round, or where's that? What's the status of that?
Yeah, so we're always actively raising.
Every early stage biotech is usually open for receiving funds.
Yeah, so we've done a lot of smaller friends and family rounds, in that $500,000 to a couple of $1 million. Unfortunately, first in human study is going to be 10 to 15 million, to get it into patients. Again, our approach is to obviously keep the company financed while we look for the larger investment that'll fund the clinical studies. And part of that approach is going to be, I think we're looking to list on a US stock exchange, either NASDAQ or the New York Stock Exchange, and raise some funds there. Being in the US, like I said before, will allow us to access a greater pool of capital, that has interest in the in vivo CAR space or the in vivo immune reprogramming space. And I think if we tell the story enough, we'll get some investors who understand and are willing to take the chance to see if the clinical data proves out. Yeah,
from a strategic perspective, you said that makes sense. You're moving from the Canadian to look at potentially to the NASDAQ to get more exposure to the US capital markets.
Exactly, yeah.
Where can people find you or keep up with what's going on at ME and what's next?
Our website is just metherapeutics.com. We post all our news and investor decks, and our socials are all there. So that's probably the easiest way.
Perfect. And then I know it's an acronym for something. What is ME stand for?
Right. So ME stands for myeloid enhancement. So the idea was, and everything we do is targeting towards myeloid cells, which are, like I said before, these immune cells that make up a large part of the tumor microenvironment and shape the anti-cancer response there. And so our focus has always been on the tumor microenvironment and manipulating that to create a strong anti-cancer immune response. And that's kind of where the name originated.
Perfect. Well, thank you so much for your time. I appreciate it. I look forward to following your journey and seeing you on the US exchange.
Yeah.
And then seeing some trials in human patients.
Yeah, we're excited. And thank you for having me.
Yeah, thanks so much.
Okay, take care.
You've been listening to Stories in Life Sciences, sponsored by MedAccess AI, where we explore people, ideas, and breakthroughs shaping the future of biotech and healthcare innovation. To learn more, visit medaccessai.org and subscribe on YouTube to watch the full conversations and new episodes as they launch. I'm Christopher Wilson. Thanks for joining us. Until next time, Keep shaping the future, one story at a time.